We are now in an era where, in addition to the normal traditional electron microscope suite, and as you know with Zeiss, it's always been a unique opportunity to merge the data sets from light microscopy and electron microscopy together. But this marks the first year really when we're fully integrated, including the X-ray microscope suite as well. And I think what you're now seeing really for the first time ever is the realization of something a number of us have been trying to work on for the last 5 or 10 years, which is the actual ability to measure something non-destructively in 3 dimensions with your X-ray microscope suite, and that includes over time, meaning in environmental conditions, loading the sample, looking at microstructural evolution, but then being able to take the richness of that data set, actually load it into the electron microscope, excuse me, in particular into the fib, and actually have the ability to direct where you're going to cut with the fib. So you can now do localized microscope imaging at any position inside that volumetric area, exactly on the defect you want. And so as a microscopist, that's exciting, of course, because it's technically very interesting and tricky. But I think as a material scientist is where it's the most interesting, because we now have the ability to go back up from these length scales we've been involved in the last 10 years at racing for the atom and the angstrom and these wonderful TEM studies, now back to a length scale, which frankly is the one that's been the origin of material science in the first place, which is microstructural defect evolution, corrosion studies, reliability and fracture studies, and now actually be able to unlock the most important part, which is what were the initial conditions of the microstructure in that sample before you actually made changes to it. So you can envision now an opportunity to measure a sample in 3 dimensions, characterize the microstructure, whether that's the grain structure, defects, corrosion pits, right? And then actually evolve that microstructure over time, continually imaging that in 3D, but then of course be able to zoom in on exactly the defects you care about, cut open into that with a fib, and now use this whole wide range of electron and light microscope techniques to really look at elemental composition and really high resolution imaging. On the X-ray side, it's been really the ability to get high resolution images while the sample is in the presence of a chamber. So these could be a load cell, this could be the ability to heat a sample while you actually have it in situ. So that involves a new kind of X-ray optics that allow you to have high resolution, still while the sample is a relatively large distance from the source. But of course, technically that also means we have to have very good stable rigs for handling this, right? And then the ability to analyze this in software. I think the next thing that was important is the ability to have software that can handle these very large data sets, comparing images that are taken over time, but also to be able to now navigate from the X-ray image all the way into the electron microscope image. So programs like Atlas V are ones that are going to actually allow you to do this seamlessly with one operator being able to use both systems. And then of course the third part is you have to have this beautiful range of electron microscopes, and I think we can really leverage off of maybe the last 10 years or so of developments and things like these newer high current fibs that are coming out on the Zeiss crossbeam, as well as all of the wonderful ability to do correlative microscopy and compare all the data after the fact from all these different modalities together.
And are there any life sciences applications that will benefit from this technology?
Yeah, so there are, and I think the ones we tend to see on the X-ray side involve things like environmental toxicology studies, looking at nanoparticle uptake into animals, looking at embryology and looking at early stage embryo phenotyping, and actually being able to look at the entire organism and zoom in and out to the different subregions of interest. And frankly, there's a growing amount of interest now in looking at the interaction between cell cultures and the tissue scaffolds, right? Everybody realizing now, of course, that whether it's a 3D or a 2D scaffold makes a pretty big difference on the way the culture grows. So that means now, how can we look at this culture in 3D and then decide where we want to go zoom in later?